Chapter 16 gives us a brief introduction of how DNA came to be seen and accepted as the molecule that contains our genetic information. It also has a section that tells us about the structure of the DNA molecule, from the structure of the double helix to higher levels of structure to the level of the chromosome. And then there is a part that tells us about the process of DNA replication. We remember from chapter 5 that DNA is a nucleic acid. DNA and RNA are both nucleic acids. The building blocks of nucleic acids are nucleotides, and nucleotides are themselves a complex molecule that consists of 3 different parts, and we're circling a complete nucleotide here. The 3 parts of a nucleotide include a 5-carbon sugar. In the case of DNA, that 5-carbon sugar is called deoxyribose, a phosphate group, and a nitrogenous base. The phosphate group and the sugar in all DNA nucleotides are the same, but the nitrogenous bases, there are 4 different ones, and the names of these bases are thymine, adenine, cytosine, and guanine. The first part of the chapter describes several very important experiments that established DNA as the carrier of genetic information. And they were done in the 20th century, some early in the 20th century, some a little bit later. They include the transformation experiment by Griffith, the follow-up of that experiment by Avery, MacLeod, and McCarty, the T2 experiment, sometimes called the blender experiment by Hershey and Chase, the determination of the pairing rules by Chargaff, and the elucidation of the structure of the DNA molecule as a double helix by Watson and Crick, with a very important contribution by Franklin. During the beginning of the 20th century, DNA's reputation was basically that of a structural molecule that is there to give support to the proteins in the chromosomes. In other words, the proteins were thought to be the information-containing molecules, and the DNA was supposed to be like a scaffold that held the proteins together on the chromosomes. And the reason for that was that proteins are much more complex molecules. Since there are 20 amino acids in proteins and there's only 4 DNA nucleotides, so people thought that DNA was just too simple a molecule. In 1928, Frederick Griffith published an experiment in which he observed something interesting going on that eventually led to the acceptance of DNA as the carrier of genetic information. Frederick Griffith was experimenting with a bacterium called Streptococcus pneumoniae, and he was working also with mice. So Streptococcus pneumoniae causes pneumonia, an inflammation of the lungs, in mice. He had 2 strains. The 2R strain was a non-virulent strain. In other words, if he infected the mice with 2R, they would not die. But if he used the 3S strain, that was a virulent strain, the mice would die. And then it was known that the virulent strain had a polysaccharide coat, so like a carbohydrate sugar coat that covered the bacterial cells. And the letters R stands for rough and S for smooth, and that referred to the shape of the bacterial colonies in a petri dish. So his observation was that live 2R bacteria could become transformed into live 3S, so one strain transformed into the other, when he injected in the same mouse live 2R bacteria mixed with dead 3R ones. He never identified himself what the transforming principle was. He was killed during World War II, so the work that he started was finished by some other investigators. Their names were Avery, McLeod, and McCarty, and we will hear about their experiment in the upcoming slides. So let's take a look at the actual transformation experiment that Griffith did using the Streptococcus pneumoniae. If he injected the 2R strain, that was the non-virulent one, if he injected live 2R bacteria into the mice would be all right. They might get pneumonia, but they would get better again, and he would find no bacteria in their blood later. If he injected live 3S bacteria, the virulent strain, the mice would get pneumonia and they would die in a few days, and when he did an autopsy he would find live 3S bacteria in their blood. If he first killed the 3S bacteria, like if he boiled the bacteria and then injected them into the mice, the bacteria would be dead and the mice would be okay, which was sort of expected. You know, the bacteria are dead, they won't kill the mouse, and there would be no bacteria in the blood of the mouse either. In another group, he injected a mixture of the 3S that were heat killed, that didn't kill the mice before, and the live 2R that didn't kill the mice either. When he injected the mixture of those two into the mice, he found the mice actually did die. And when he looked at their blood, when he looked in an autopsy at their blood, he found live 3S bacteria in their blood, even though he never injected live 3S into those mice. So there's that transformation principle. The live 2R was transformed into live 3S. But like we said before, Griffith did never himself identify what the transforming principle was. Here is the illustration of the experiment that we just were talking about. So when Griffith injected a mouse with the living S strain bacteria, the living S Streptococcus pneumonia, he found that they developed pneumonia and they died. If instead of using the S strain, he used the R strain and injected living cells of the R strain into the mouse stayed alive and healthy. If he used the S strain, but before injecting the bacteria into the mouse, he killed the bacteria by boiling them, then the mouse would stay healthy also. He was injecting dead bacteria into them. And now in the fourth group, he mixed the dead virulent strain, the ones that were boiled, with the live non-virulent strain, the R cells. So dead S plus live R. He injected them together into a mouse, and the poor mouse died. And when he looked at the blood of the mouse after the mouse died, he found living S strain cells in the blood. Now that was odd because he never injected living S cells into the mouse. He injected dead S and living R. So that's the transformation part. The transformation is that the living R cells were transformed into living S cells. And this happened over and over every time the experiment was repeated. But Griffith himself didn't know what the transforming principle was. So that part was left to Avery, MacLeod, and McCarty to discover. Avery, MacLeod, and McCarty were able to identify what the transforming principle was in Griffith's experiment. They published their results in 1944. They were working with an extract of the 3S virulent strain bacteria that had the same capability of transforming cells as the boiled S3 or 3S bacteria themselves. They separated this extract into 3 groups, and they treated these 3 groups with 3 different types of proteins that each inactivated a different macromolecule in the extract. So they knew that the complete extract would have DNA, and it would have RNA, and it would have protein. If you want to figure out which of those 3 molecules is the active agent, then if you say, well, if I break down the protein and after that the extract doesn't work anymore, that would point to the protein being the active agent. If I break down the RNA and that makes it not work, then it would be the RNA. If I break down the DNA and that makes it not work, it would be the DNA. So they did 3 separate experiments. In one experiment, they combined the extract with protease, which can inactivate proteins by breaking the peptide bonds between amino acids. In another group, they mixed the extract with a protein called RNase, an enzyme that can break down RNA specifically. And in a third group, they mixed the extract with an enzyme called DNase, and DNase inactivates DNA specifically. So after they did that, they mixed those treated samples separately with live 2R bacteria, and they checked in which of these is transformation inhibited. And when they looked at them, they saw, well, in the sample that was treated with protease, transformation was just happening as normal. In the one that was treated with RNase, transformation happened as normal. But in the one that was treated with DNase, transformation stopped. And that pointed out that DNA had to be the transforming principle, because inactivating DNA stopped transformation. Here is a picture that I found on the internet that shows the Avery, MacLeod, and McCarty experiment. The parts that they talk about in our book are over here, where the extract was treated with RNase or protease or DNase. Here they also talk about treating different samples with lipase and a an enzyme that would break down carbohydrates, say amylase, they call it carboase. The idea is that they're saying that they used various enzymes to inactivate specifically all the macromolecules that they could think of. And then what they found was that the only macromolecule that when it was inactivated would prevent transformation was DNA. So the only treatment that prevented transformation was DNA's treatment. Now we're going to accumulate even more evidence in favor of DNA as the carrier of genetic information. This experiment was published in 1952, and it was performed by Alfred Hershey and Martha Chase. And they were working on a very interesting system using a bacteriophage called T2. Bacteriophages are viruses that specialize in infecting bacteria. And this particular bacteriophage was used to infect E. coli bacteria. What makes using bacteriophages for this experiment so appropriate is that it's a very simple system. A bacteriophage just consists of a protein coat or capsid that surrounds a DNA core. That's the only thing. It doesn't have any fats, it doesn't have any carbohydrates. It has a DNA core surrounded by a protein capsid. They found in their experiment, so these are just their general results, that only the DNA part of the bacteriophage was entering the cells during the infection. They observed that when new viruses were produced during an infection, they would have both protein and DNA. From that you can conclude that the DNA must be coding for the new protein capsids in the progeny viruses, because no protein ever went into the cells, yet protein plus DNA was coming out of the cells, so the DNA must have contained the information to code for the new proteins. This slide shows us an electron micrograph of the T2 phages infecting a bacterial cell. So the brown part here in the bottom, is the bacterial cell, and there are 3 phages sitting on top of it. Isn't it interesting, that shape? It looks so alien, almost like a machine, but that is the shape of these phages. This capsid is made of protein, and inside the capsid, in here, is a DNA molecule. And in this picture we actually can see the DNA molecule entering the bacterial cells, but Hershey and Chase didn't know that at the time. Let's take a look at Hershey and Chase's experimental setup. They used 2 different radioisotopes. They used phosphorus 32 or P32 to label the DNA of their T2 bacteriophages, and they used sulfur 35, S35, to label the protein of the T2 phages. The reason for choosing these particular radioisotopes is that phosphorus can be found in DNA, in all the phosphate groups in each nucleotide, but there is no phosphor in proteins. In the same way, sulfur can be found in proteins in some of the amino acids like cysteine and methionine, but there is no sulfur at all in DNA. That means that if you're labeling your phages with P32, you can be guaranteed that the label is only going to be incorporated in the DNA. There will not be any cross-labeling or any contamination of label going into protein, and vice versa. The S35 will only be going into protein. So they had very specific labels for protein and specific label for DNA. So they had a batch of phages that were labeled in their DNA with P32 and another batch that was labeled in their protein with S35. They mixed these phages with E. coli in separate experiments. So they allowed the labeled phages to attach to the E. coli cells and sit there for maybe 5-10 minutes. Then they put those labels, they put them through a blender. So put them in the blender, press frappe or mix or blend, whatever. The action of the blender won't hurt the bacterial cells or the phages, but it will shear off anything that is on the outside of the bacterial cells. So in other words, anything that's sticking out from the cells is going to be separated from those cells. And after they did that, they centrifuged the mixes separately. So they centrifuged the mix that had the P32 labeled phages, and they centrifuged the mix that had the S35 labeled phages. And when they looked at their results, they found that when they had used P32 to label the phages, they could find the label in the pellet of the tube together with the bacterial cells. If, on the other hand, they had used S35, then the label would be found in the supernatant, which is above the bacterial cells. They also noticed that some of the progeny phage that they got, if they let that infection continue and lyse the bacterial cells and produce progeny phage, some of those progeny phage had P32 in their DNA, but none of them had S35 in their protein. Let's go to the next slide so we can see what those tubes looked like, and I can show you what a pellet looks like and the supernatant looks like. So here is the experiment, and it's color-coded. The S35 is the pink group here on the top, and the P32 is the blue group. And you see their procedure. They have the starting here on top, they have the S35 labeled phages that are allowed to attach to bacterial cells. So that we see here, the phages are sitting on the surface of the cell, and the protein is labeled. So the protein is pink. Then they put that whole stuff, that whole culture, through the blending process. So they briefly put it through the blender here, and that is going to separate anything that wasn't inside the bacterial cells. When they now put that mix through the centrifuge, they obtain the result, and here is the tube with the centrifugation, and they find that they get a pellet and they get a supernatant. The pellet contains the bacteria, but there is no radioactivity with the pellet. They find that all the radioactivity is found in the supernatant. The supernatant is the liquid that is floating above the pellet. And if you look at the interpretation, is that the pellet contains the bacteria with the DNA of the phages, but because the DNA wasn't labeled in this experiment, there is no label with the pellet. and the supernatant contains all of the labeled capsids that were sheared off from the bacterial cells. Let's look at the other experiment. Here, in the second experiment, they labeled the DNA of the phages with P32. So look now, you see that the capsids are gray, they're not labeled. What the inside of the capsids, inside is that core of DNA that is blue, and you can even see here this one that the DNA is going into the cells. They do exactly the same procedure. They let the phages attach to the cell and sit for a few minutes, and then they put that mix in the blender and they blend it, and we see here the same thing is happening. Anything that's outside the bacterial cells gets sheared off. But now, what is inside the bacterial cells, the DNA, has the label, while what got sheared off does not have the label. So let's look at the results after the centrifugation step that comes after the blending. Again, we get a pellet at the bottom. The pellet basically contains all the dense material, the big things. And then we have a supernatant with just like the liquid and things that are very light in density. The supernatant again contains the protein capsids that were sheared off, but this time there is no label, no radioactivity in the supernatant because the proteins were not labeled in this experiment, in this group. On the other hand, when they used P32, they found the label in the pellet associated with the bacterial cells. And the interpretation for that was that exclusively the DNA of the phages was entering the bacterial cells. In both of the groups, in the groups in which the protein was labeled and in the groups in which the DNA was labeled, only the DNA was entering the bacterial cells. But when you then centrifuge, when you do the blending and the centrifugation, you find that if you label the DNA, then you will find it back inside the bacterial cells. If you labeled the protein, you will find it in the supernatant, because the bacterial cells are much denser than those protein capsids of the phages. So the protein capsids of the phages stay in the supernatant, while the DNA of the phages, which in itself is very small and very, very light, but when it's trapped inside the bacterial cells, it's going to go down into the pellet, and it will stay associated with the bacterial cells. So let's review the conclusions from the Hershey and Chase experiment. First, the P32 label was found in the pellet together with the bacterial cells. Conclusion: the T2 DNA had entered the bacterial cells. The only way that that light DNA molecule can be found with those heavy bacterial cells is if that DNA molecule was located inside the bacterial cells, and therefore it got pelleted with the bacterial cells. Result: the S35 was found in the supernatant. Conclusion: the T2 protein, the protein part, had not entered the bacterial cells. In other words, the protein capsids never entered the bacterial cells. When a virus infects a bacterial cell, it injects, or at least the T2 virus injects its DNA into the bacterial cells, but the capsid just stays outside. So the capsid, during the blending procedure, got sheared off of the surface of the bacterial cells, and then during the centrifugation procedure, these light capsids remained suspended in the supernatant and did not go into the pellet with the bacterial cells. Third finding, they noticed that some of the T2 progeny, in other words, the next generation of viruses that were formed from these same bacterial cells, if you allowed that infection to proceed, eventually the bacterial cells would lyse, they would burst, and they would release a whole bunch of phages, each of them. So they would release the next generation of phages, the progeny phages. Some of these progeny phages had P32 in their DNA. They were labeled with P32. None of the progeny phages had S35 in their protein. So the P32 but not the S35 was found in the T2 progeny. What that points to is that the DNA was physically being passed from the infecting T2 viruses to their progeny. For a DNA molecule to make it into the T2 progeny viruses, it means that that DNA molecule had to be inserted into a new virus particle. And then also, the protein was never being passed on physically, because they never found labeled protein with the progeny phages. The other thing that they could conclude was that the DNA had coding capacity. Why? Well, only DNA had entered the bacterial cells, but complete variants were coming out that have both DNA and protein. Since no protein had entered the bacterial cells, in order for protein to come out of the bacterial cells during the infection, like when the bacterial cells burst and they release the new phages, that new protein had been coded by the DNA that entered the bacterial cells. So the DNA contained the genetic information that coded for those protein capsids in the progeny phage. This slide shows us a series of data and characteristics that were found about DNA that came to be known as Chargaff's rules, because they were discovered by Erwin Chargaff. So the first thing was that he found that each species has a characteristic percentage of A, C, G, and T nucleotides. So for instance, in humans, we have 30% A nucleotides, while E. coli has only 25% A. And if you look at different humans and different E. coli bacteria, you find that in general, E. coli has 25% A, while humans have 30% A. Then another point was that even though each species has their own characteristic fraction of A, C, G, and T, in all of these organisms, the percentage of A is the same as the percentage of T, and the percentage of C was the same as the percentage of G. In other words, coming back to the humans that have 30% A, if the percentage of A is the same as of T, then it means that we have 30% A and also 30% T. If you want to calculate the percentages of the other two nucleotides, and you would subtract the 30 plus 30 from the A plus T from 100%. So that would be 100% minus 60%, leaving 40% for G plus C. And that would give us 20% each for G and 20% for C. In E. coli, it has 25% A, so that would mean 25% A, 25% T, and then G and C would be 50 together. Each of them would also be 25%. That's just by coincidence. E. coli appears to have about equal percentages of all the 4 nucleotides. The conclusion, though, that you can come to from the fact that the percentage of A is always the same as the percentage of T, and C is always the same as G, is that A pairs with T and G pairs with C, which is indeed what happens. A pairs with T through 2 hydrogen bonds. So the A base in the nucleotide pairs with the T base in a nucleotide on the opposite strand, and a G base pairs with a C base. GC pairing has 3 hydrogen bonds. Another very important discovery was that of the structure of the DNA molecule. The DNA molecule has a double helical structure that was published for the first time by Watson and Crick in 1953. They used an X-ray diffraction photograph that was made by Rosalind Franklin, who was an expert in X-ray diffraction. She worked down the hall from them, and her boss, Wilkins, showed the picture to Watson, who immediately realized its significance and what it meant with respect to the shape of the DNA molecule. About 10 years later, Watson, Crick, and Wilkins shared the Nobel Prize for Chemistry for this discovery. Unfortunately, at that time, Franklin had been dead for about 5 years. She died of ovarian cancer. This picture shows Watson and Crick with a model of the DNA double helix, and this was taken shortly after they published the paper, or at the time when they were getting ready to publish the paper. So it shows the double helix with the backbone of sugars and phosphate groups on the outsides, and then the bases stacked on top of each other in the middle, connected by hydrogen bonds. Let's review the structure of the DNA molecule again here. DNA is a nucleic acid. RNA is also a nucleic acid. And the building blocks of all nucleic acids are nucleotides. A nucleotide is a composite molecule that consists of a phosphate group, such as we see here, a sugar, in the case of DNA, it's a deoxyribose sugar, and a nitrogenous base. There are 4 different kinds of nitrogenous bases: thymine, adenine, cytosine, and guanine. And the nucleotides on a single strand, so as we see here, connect to each other through covalent phosphodiester bonds. And a phosphodiester bond is basically the connection between 2 sugars and a phosphate group, phosphodiester. But these days it has also come to mean just the sugar phosphate bond that occurs over here. So each sugar phosphate connection is considered a phosphodiester bond. And here we see the double helical structure of the DNA molecule. It forms a spiral, and this part in the middle shows us how the 2 strands in the double helix connect with each other through hydrogen bonds. 2 hydrogen bonds between an A and a T, and 3 hydrogen bonds between a C and a G. But you can see how these bases, like this C, is part of this nucleotide here, and this T is part of that nucleotide there, and so this T is part of this nucleotide. So each nucleotide has 3 parts: a phosphate group, a sugar, and a base. And here we see a representation of the DNA molecule. Is basically the same information as this one, but it's called a space filling model. And that gives us maybe a slightly more realistic view of what the actual shape of the atoms in the molecule are. An interesting fact about DNA is that the two strands in the double helix are anti-parallel to each other. In other words, they are oriented in opposite directions, as you could say that they're upside down with respect to each other. The two strands are also complementary to each other. In other words, the two strands are not identical but complementary, and each has the information to give us what the identity of the other one is. So if you know what the bases are on the strand on the left side, then you can immediately write down the bases of the strand on the right side by using Chargaff's pairing rules, right? You know that A pairs with T and G pairs with C. So if you see an A on the left side, you know there should be a T on the right side opposite that A. The illustration shows us both the anti-parallel nature and the complementarity of the two DNA strands in a double helix. The anti-parallel nature can be easiest observed by looking at the sugar molecules. So these are the deoxyribose sugar molecules in 4 nucleotides on one side of the double helix. Notice that the oxygens are pointing up in all of them. When we go to the opposite side, here, you notice that all the oxygens are pointing down. So one side is oriented in the opposite direction of the other side. So let's say if we're putting an arrow this way for one side, then it's going to be that way on the other side. Another thing is that each nucleotide will have a 5' end and a 3' end. The 5' end of a given nucleotide is at the phosphate, and the 3' end is at the deoxyribose. In every DNA molecule, you're going to have one free 5' and one free 3' end. Here is the free 3' end of this side, and you see here is the deoxyribose, and this is the hydroxyl molecule at the 3' end. On the other strand, on the right side, the 3' end is going to be here, and the 5' end there. So that also shows that they're anti-parallel to each other. And then the complementarity, wherever you have a T, there's going to be an A opposite that. Wherever you have a G, there's going to be a C, and so forth. The fact that the two strands in the double helix are complementary immediately suggested a mechanism for how DNA replicates itself. Watson and Crick said that themselves when they published the structure of the DNA molecule. Each of the two strands has the necessary information to specify the other strand. So that means that during the process of DNA replication, the two strands can separate from each other, and each of them can become the template for a new complementary strand. And using the pairing rules that Chargaff discovered, you just pair A with T and G with C, and then you can regenerate a new strand opposite each of the two strands that separated from each other. So this is a model that proposes a way or a mechanism for how DNA could replicate. Here you have a parental DNA double helix, two strands that are binding to each other through hydrogen bonds. The two strands separate from each other, and each of the two strands, notice that the parent strands are dark blue, each of the two strands becomes the template for a new strand that is complementary to it. So this one here on the left, this one, got this partner, and the one that was on the right got that partner. When you now compare these two strands, these two double helices with each other, you find that they are identical, and they're also identical to the double helix that they came from. So you have replicated this double helix and made two identical daughter molecules, each of which is going to have one new DNA strand, light blue in this case, and one old DNA strand, dark blue. The model that we just talked about is known as the semi-conservative DNA replication model. It was the one that was suggested by Watson and Crick in 1953. They said that the two DNA strands will separate during the process of DNA replication, and each of the strands becomes the template for a new complementary strand. Then after the process of replication is finished, there's going to be two identical double helices. Each one is going to have one new and one old strand. And the two old strands that were the templates, they will not come back together again because each of them will have a new partner. Each of them is conserved, like the sequence is completely conserved, but they're not conserved together. That's why the model is called semi-conservative replication. As soon as Watson and Crick published their paper and talked about how they thought DNA replication could occur, they suggested the semi-conservative model. There were two other models that were also proposed by other people that were known as the conservative model of DNA replication and the dispersive model. According to the conservative model, the two original DNA strands that serve as templates during the process of replication do come back together after replication is finished, and they're conserved together as a unit. That's why that was called conservative replication. And then the other model for dispersive DNA replication suggested that the two strands of the double helix are each fragmented. They're going to be like broken up during the process of DNA replication, and then DNA repair fills in the gap between the strands using both old and new strands as a template, and neither of the two strands is going to be conserved. So that's why it was called dispersive. At the time, each of these three models might have seemed a plausible model, and since you couldn't disprove either of them, all three of them were valid hypotheses. It took until 1958 for an experiment to be done that gave conclusive evidence in favor of the semi-conservative model. Here is a picture of the three competing models for explaining DNA replication. The conservative model on the top shows us a parent cell, so you have a double helix. Notice that both of the strands are dark blue. After the first replication, it would predict that you would get two molecules, one that is exactly like the original with both strands dark blue, and then the second molecule has all new nucleotides, so it is light blue. In the second replication, each of these two from the first replication replicate. So now you've got the original molecule that gets passed on intact, it's the one that was made from it, and the two that were made from the first new molecule. So in the end, you see that generation through generation, you keep passing on the same original DNA molecule from one generation to the other, intact, with the same two strands attached to each other. The semi-conservative model is different. According to the semi-conservative model, the two dark blue strands that we're looking at here separate from each other, and each of them serves as template for a new strand, which is light blue. So now in this second generation, each of the two molecules generated is going to have one old dark blue and one new light blue strand. And if you can follow the fate of the original dark blue strands in the second generation, here is still a molecule that is half old, half new, and here also one half old, half new. And these are all new. Then finally, the dispersive model says that from that original double-stranded, double helical DNA molecule, both of the strands are going to get broken in different places, and then DNA repair is going to fill the gaps so that you get two new double helices, both with old and new parts to them. And the same thing happens in the second generation. It's just that the old DNA is going to get progressively smaller as a fraction of the total. Now, without going into the details of the experiment that was done by Messelson and Stahl, we can say that it provided conclusive evidence in favor of the semi-conservative model of DNA replication. It basically supported all of the predictions of the semi-conservative model, and it eliminated both the conservative model and the dispersive model as possible ways in which DNA can replicate. The fact that the conservative model and the dispersive model were both eliminated by this experiment does not make them bad scientific hypotheses. On the contrary, a good scientific hypothesis is one that makes predictions that can be tested, even if eventually that means that the hypothesis may have to be abandoned if the results of the test don't support the hypothesis. DNA replication is semi-conservative in all organisms. Using E. coli as a representative of prokaryotic organisms, we find a circular chromosome in E. coli that contains practically all of its DNA, and that one circular chromosome has 4.6 million base pairs. There is one single origin of replication on that chromosome, so replication begins at just that location. Since it is bidirectional, we'll find two replication forks at that origin, and the replication fork is the place where the two parental DNA strands are going to get separated from each other, and we'll see replication forks in upcoming slides. Compare that with an organism such as the human, a eukaryotic organism, and we have not one chromosome, we have 23 different kinds of chromosomes, and we have two copies of each chromosome, so we have actually 46 chromosomes in each of our cells, and those chromosomes are not circular but linear. All of our DNA together in all the chromosomes encompasses about 6.4 billion base pairs, so more than 1,000-fold the amount as we find in E. coli. The challenge is now how to replicate so much DNA in a reasonable amount of time. E. coli can replicate its one chromosome in less than an hour. But if we had to replicate all of our DNA with just one origin of replication, it would take us months to replicate our DNA. And DNA replication takes maybe 10 hours. So the problem is solved by eukaryotes by having many origins of replication for each chromosome. Some chromosomes even have thousands of origins of replication. That means that replication is going to be starting simultaneously at a lot of locations, and that is going to make the process go much faster. Even though eukaryotes, their speed of replication at a given origin of replication is only about 1/10 of that of prokaryotes. I think it's 50 nucleotides per second in eukaryotes, while it's about 500 nucleotides per second in prokaryotes. Similar to prokaryotic replication, replication in eukaryotes is also bidirectional, so we will see 2 replication forks per origin of replication. We just mentioned that in bidirectional DNA replication, we get 2 replication forks, 2 per origin of replication. And in circular DNA, we see a little detail of that process. There is a single origin of replication that is shown here and here. So this here is the origin of replication. And there are two replication forks shown by the red arrows. So one is here and another one is there. And replication is going to go in both directions simultaneously from the single origin of replication. Compare that to eukaryotic DNA, in which each chromosome is going to have hundreds of origins of replication. Here we're looking at just 2 of them. Each of those 2 is very much like the single origin of replication that we see in prokaryotes. We see here is, for example, the origin of replication of this one, and there are 2 replication forks, one here and one there per origin. For this one over here, we've got 1 replication fork here and another replication fork there. Remember, the replication fork is where the 2 DNA strands separate from each other in the process of DNA replication. This slide shows basically similar information as the previous one. It also shows us a little electron micrograph of the process of DNA replication in both prokaryotes and in eukaryotes. So in prokaryotic DNA, it's just 1 origin of replication, and the process proceeds bidirectionally from there. And in eukaryotes, there are multiple origins of replication, and there are 2 forks for each origin. For the DNA to be replicated, it has to be unwound first. And there are several enzymes that are active on an ongoing basis to help that happen. Helicase is an enzyme that unwinds DNA, that separates the 2 strands from each other by breaking the hydrogen bonds between them. And helicase is basically the enzyme that maintains the replication fork. Another enzyme called topoisomerase is not directly going to unwind the DNA, but it permits the ongoing unwinding because it relieves the strain produced by the unwinding. If you envision that the unwinding is like separating two halves of a rope by just pulling them apart, then you can just imagine how in front of you the rope is going to get tighter and tighter and eventually gets tangled. The same thing happens with the DNA. What topoisomerase does is that it breaks the phosphodiester bonds on each single strand, but it can make double-stranded breaks, so it breaks both single strands. It allows the DNA to swivel to relieve that torsional tension, and then it rejoins the broken ends together, and that is done multiple times in the process of replication. Another group of proteins that allows this process to continue smoothly are the single-strand binding proteins, abbreviated SSBPs. The single-strand binding proteins stabilize single-stranded DNA. They literally bind regions of single-stranded DNA and prevent the two strands from snapping back together. The natural state of the DNA molecule is for the two strands to be attached to each other and connected through hydrogen bonds. If you separate them, they will naturally come back together again. So the single-stranded molecules are there to keep the DNA single-stranded until the process of replication is accomplished locally. And then we have primase. Primase is a molecule that synthesizes RNA primers that are used to start the process of DNA replication at each origin of replication. We're going to see that in a little more detail coming up in a new slide. Here are the proteins that we just talked about, shown at their respective positions at the replication fork. So helicase is the green one, and helicase is breaking the hydrogen bonds between the two parental strands. So it's basically separating the two strands from each other. The single-strand binding proteins are binding to the single-stranded regions of the DNA, keeping them single-stranded and keeping them from snapping back together. Topoisomerase, this big blue one over here, is basically ahead of the replication fork in the part that is still double-stranded, cutting the two strands, allowing them to swivel and relieving the torsional tension. And then primase, this pink molecule here, a big protein, is going to lay down primers, and the primers are going to be used to then add to synthesize new DNA molecules. The protein that puts down new nucleotides, in other words, that allows the new DNA strands to grow, is called DNA polymerase. In E. coli, specifically the one that does most of the DNA synthesis, is called DNA polymerase 3. So DNA polymerase adds new nucleotides, but it needs a pre-existing strand to add to. That strand is called a primer. The primer, as we already saw, is going to be laid down by the enzyme called primase. DNA polymerase adds nucleotides exclusively to the 3' hydroxyl end. That's one of the characteristics of all DNA polymerases. They can only add to a 3' hydroxyl end. It uses as template the complementary strand. When it puts down each nucleotide, when it inserts each nucleotide, it cleaves two phosphate groups from the nucleotide to provide the energy necessary for the insertion reaction. And then it hydrolyzes the pyrophosphate that is produced to inorganic phosphate. The speed at which this happens in prokaryotes, in other words in bacteria, it occurs at about 500 nucleotides per second, and in eukaryotes, for example in human cells, about 50 nucleotides per second. So not nearly as fast, but still a very impressive 50 nucleotides per second. Here's an illustration of the process of the addition of a nucleotide to a growing DNA strand. The template is the dark blue one, this one, and the new strand is the light blue one, and we're adding a new nucleotide over here. That nucleotide is going to have a T base, and the A was the template that is used for adding it. That T is a deoxyribonucleoside triphosphate. In other words, there are 3 phosphate groups attached to the sugar part. DNA polymerase cleaves off 2 of those 3 phosphate groups, and here they are, forming pyrophosphate, and then it breaks the pyrophosphate down into individual inorganic phosphate molecules, and that is an exergonic reaction that allows the insertion of that new nucleotide into the DNA molecule. So the nucleotide, a mature nucleotide that's in a DNA molecule, has only 1 phosphate group, but the precursor that is used to add a nucleotide has 3 phosphate groups, and we cleave off 2 of the phosphate groups to provide the energy for the reaction. We mentioned earlier that the 2 strands in each double helix are anti-parallel to each other, so they're facing in opposite direction to each other. That also means that each new growing strand is going to be anti-parallel to its template strand. Another fact is that DNA polymerases are only capable of adding nucleotides to a 3' end. What this means is that at each replication fork, such as this one that I'm circling here, one of the new strands is going to have its 3' end facing the replication fork, the lower one in this case. That one is going to be called the leading strand because it can be replicated without a problem, smoothly and continuously. The other strand, though, is going to be facing with its 3' end away from the replication fork, like we see here. And it's going to have to be replicated in little fragments. That strand is known as the lagging or discontinuous strand. Let's take a look at the leading strand a little more specifically. So at each replication fork, there will be 1 leading strand. It is also called the continuous strand, and it faces the replication fork with its 3' end. This leading strand is going to need only 1 primer per origin of replication. And here is a picture illustrating the leading strand in a replication fork. In the upper drawing, we're looking at a replication fork. Remember, that's the part where the 2 strands come apart. And here, we're making the leading strand, which is the upper strand. An RNA primer has been put down, and that are the red nucleotides over here. And DNA polymerase 3 is here adding DNA nucleotides to that primer, always adding to the 3' end. When DNA polymerase 3 adds a nucleotide, it produces a new 3' end with each nucleotide that it adds. So when it adds a nucleotide to the 3' end of the last existing nucleotide or primer, it puts in the nucleotide in such a way, it puts it 5' end first, and then the 3' end is free. The next nucleotide is also put in 5' end first, and the 3' end is sticking out. So we see here there is a 3' end DNA polymerase 3 keeps adding new nucleotides to that 3' end. In the meantime, helicase keeps unwinding the two template strands at the replication fork, so the replication fork is moving to the left. And that's what we see here. The replication fork has moved to the left, and the new strand has continued to grow continuously with just that one primer that was put in originally, and therefore this strand is called the continuous strand, and we'll see compared to the other one why it is called the leading strand also. Now we're going to take a look at the lagging strand of the replication fork. The lagging strand is the other daughter strand, and it faces the replication fork with its 5' end. In other words, its 3' end faces away from the replication fork. Why is this important? It is important because DNA polymerase can only add nucleotides to a 3' end, so the strand can only grow away from the replication fork. It is called the lagging or discontinuous strand, and it needs to be made in little fragments that are called Okazaki fragments. Each of those Okazaki fragments is going to have their own primer, and then DNA polymerase 3 is going to add nucleotides to that primer. Okazaki fragments are about 1,000 to 2,000 nucleotides long in prokaryotes and about 100 to 200 nucleotides long in eukaryotes. And remember, each individual Okazaki fragment is going to be growing in a direction away from the opening of the replication fork. This slide is showing us what happens in the replication of the lagging strand, the one that faces the origin of replication with its 5' end. The origin that we're going to be looking at is this one, and it is opening toward the left side, and the strand that we're looking at is the lower one over here. So all of these drawings are referring to that lower strand. In the first drawing, this one, we see how after the origin opened to some extent, remember the origin is opening in this direction, primase has deposited a primer because DNA polymerase 3 needs a primer to add to. Notice that the 3' end of the primer is facing away from the opening of the replication fork. In the second drawing, this one here, we see that DNA polymerase 3 is adding nucleotides to that primer, and it is moving towards the right, away from the opening of the replication fork. In the third drawing, we see that DNA polymerase 3 has reached the previous primer that was put down over here, and DNA polymerase 3 has detached from the template. In drawing number 4, we see that a new primer has been put down. They call that the RNA primer for fragment number 2. And we see how DNA polymerase 3 has added some nucleotides to it, and again it is moving towards the right. The replication fork is opening in this direction. So Okazaki fragment number 2 consists of the primer number 2 plus the DNA fragment that is laid down by DNA polymerase number 3. In the meantime, another DNA polymerase is coming in, DNA polymerase 1, and DNA polymerase 1 is going to remove the nucleotides of each primer, because you can't leave RNA nucleotides in a DNA strand. So DNA polymerase 1 is going to remove the RNA nucleotides one by one and substitute them with DNA nucleotides. And we see that happening here, and we see it happening here as well. So it is moving also to the right side from 5' to 3', and taking out the red nucleotides and substituting them with blue nucleotides. And then finally, in the last drawing, this one here, we see how ligase is going to fill the nicks that are left in the strand between the last nucleotide put in by DNA polymerase 1 and the one that was from the previous fragment. So there is a little nick here. That nick is going to be sealed by ligase, which forms a phosphodiester bond between the two. And at that point, once ligase has connected those two fragments, then the DNA fragment is complete. Now we're going to summarize the process of replication, looking at both the leading strand and the lagging strand. The replication fork that we're looking at is going to be the left fork from this origin of replication, and the leading strand is going to be the upper strand. We see how the replication fork is opening to the left with helicase separating the two template strands from each other by breaking the hydrogen bonds between them. And we see on the leading strand how DNA polymerase 3 is adding nucleotides to the 3' end of the growing strand, and basically it is following the opening of the replication fork, and therefore this strand is known as the leading or continuous strand. Only one primer was needed right at the opening of the origin of replication, and that was then added to all the way smoothly and until you get to the end of that replicon. Now, if we look at the lagging strand, we see that, of course, it is anti-parallel to the leading strand. So the growing strand has its 3' end facing away from the origin of replication. In other words, it is going to be growing in the opposite direction that the origin of replication opens. In this strand, the most recent events are shown closest to the origin of replication, and the most long ago are the ones that are further away from the origin of replication. If we take a look at what is happening right now, or most recently, we see that as helicase is opening the two strands, we've got the single strand binding proteins maintaining the DNA single-stranded, and then we see over here there is a primer, the red nucleotides, that is being laid down by primase. Primase is moving in the 5' to 3' direction. A little bit further down, we see another primer. That one was the one that was laid down before this one. In that one, DNA polymerase 3 is adding DNA nucleotides to the primer, the blue nucleotides, and it is going to go until it reaches the previous primer that was laid down, this one. When DNA polymerase 3 reaches that primer, it's going to detach, going to get off the template, and then it's going to come back and it's going to add nucleotides over here. So it backtracks along the DNA strand. DNA polymerase 3 also moves from 5' to 3'. A little bit further back, we see DNA polymerase 1 removing the RNA primer and substituting the RNA nucleotides with DNA nucleotides one at a time, moving in the 5' to 3' direction. So you see that the red nucleotides are being taken out and substituted for blue DNA nucleotides. And finally, even further back, we see DNA ligase linking together the last fragment that was substituted, like the last primer that was taken out by DNA polymerase 1 and substituted with DNA nucleotides, and this fragment that was made before. So DNA ligase is going to make a phosphodiester bond between each of those nicks that get left, and when DNA ligase is done linking together all of the fragments, then the lagging strand will be completed as well. Table 16.1 very helpfully summarizes all of the enzymes that are used during DNA replication. We see helicase. Helicase unwinds the double helix at the replication fork. It actually makes the replication fork as it moves along. Single-strand binding proteins will stabilize the single-stranded DNA so that it can be replicated and the two parent strands don't come back together prematurely. Topoisomerase relieves the strain, the torsional strain, also called overwinding strain, that is formed ahead of the replication fork by actually breaking the strands, letting them swivel, and letting them rejoin. Primase is an RNA polymerase that is going to synthesize an RNA primer at the 5' end of the leading strand and at the 5' end of each Okazaki fragment of the lagging strand. RNA polymerases need a template, but they don't need a primer, so they're the ones that are used to make the primers. Continuing table 16.1, we have DNA polymerase 3. DNA polymerase 3 uses the primer that was just put down by primase to add to, and it's going to use the parental DNA as template to synthesize a new DNA strand. DNA polymerase 1's job is to take out those RNA nucleotides that were previously put down by primase. So all those RNA primers have to be removed and replaced with DNA nucleotides. When DNA polymerase 1 does that, when it reaches the beginning of the previous fragment, it cannot make a phosphodiester bond between the last nucleotide it puts in and the first nucleotide of the previous fragment, because it can only hook a nucleotide to the previous nucleotide that it put in. So there is a nick or a little, yes, you can see it here, a little nick is going to be left, and that nick is going to be sealed by DNA ligase. So DNA ligase catalyzes the formation of a phosphodiester bond between two nucleotides on the same strand. These three subtopics, the proofreading and repairing of DNA, evolutionary significance of altered DNA nucleotides, and replicating the ends of DNA molecules, are not going to be on our exam. Now we're going to switch gears from the mechanics of DNA replication to an account of the different structures of DNA. So far, we've been looking at the primary and the secondary levels of DNA structure. The primary level is the order or the sequence in which one nucleotide follows another, and at the primary level is where genes have their informational content. The primary level is maintained by covalent phosphodiester bonds between the sugars and the phosphate groups on each single DNA strand. The secondary level involves the formation of the double helix, and it is what makes the double-stranded DNA molecule. It is maintained by hydrogen bonds between the two strands. The tertiary level actually has different sublevels, and it involves different amounts of folding and wrapping that we're going to be looking at in the upcoming slides. And it involves different structures also, the nucleosome and several different fibers, and finally the chromatid. DNA associates with proteins to form a structure called chromatin. Chromatin and chromosomes are actually the same thing. Chromosomes are a very condensed version of chromatin, or we could say that a very relaxed chromosome, where the DNA is dispersed, can be called chromatin. The tertiary level of DNA structure takes us from the nucleosome all the way to the metaphase chromosome in increasing levels of DNA condensation. So we have the nucleosome, a 30 nanometer fiber, a 300 nanometer fiber, and then the metaphase chromosome. The nucleosome represents the first level of DNA condensation. A DNA double helix, 146 base pairs of DNA double helix, wraps around an octamer of histone proteins to form a nucleosome. An octamer means 8, and there are 8 histone proteins, 2 each of these 4 types, H2A, H2B, H3, and H4. In general, histone proteins have in common that they tend to be rich in basic or alkaline amino acids, such as arginine and lysine. These basic amino acids interact with the acidic phosphate groups on the DNA double helix and allow the DNA and the histone proteins to interact and touch. The diameter of a nucleosome is about 10 to 11 nanometers. Compare that with the diameter of the DNA double helix, which is 2 nanometers. So you see it's gotten thicker, but it's also been condensed quite a bit because you're wrapping 146 base pairs around that nucleosome core. These illustrations show us the structure of the nucleosome. The histone octamer is shown here, and you can see H2A, H2B, H3, H4, and then here's another copy of H4, another copy of H2B. The other 2 copies of H2A and H3 would be in the back. And then you can see how the DNA is wrapped around that nucleosome core. There is another histone protein that we haven't mentioned yet that is called histone protein H1, that is known as a linker histone protein. It is not part of the nucleosome core. Here is another representation showing the nucleosome core as like a flat disc or a flat ball, and then the DNA as a rope, the double helix as a rope that is wrapping around those histone octamers. This is an electron micrograph of DNA that is associated with nucleosomes. This structure came to be called beads on a string, and you can see why. The nucleosome cores are shown here, those little balls, and then there is DNA wrapped around each of them, and you can also see that there is a little distance, there is DNA between one nucleosome core and the next. That DNA is known as linker DNA. So remember, what is associated directly with the nucleosome core is about 146 base pairs of DNA. The next level of DNA structure is achieved, the 30 nanometer fiber, with the packing of the nucleosomes together. And it involves the use of that histone H1 that we saw in the previous slide, the one that does not belong or does not form part of the core nucleosome. I'm circling one of them here. They're the little yellow beans are the histone H1s, and they serve sort of as safety pins that hold the DNA attached to the nucleosome and keep that wrapped up. When these H1 histones interact with each other, then the nucleosomes coalesce onto a denser fiber which has a diameter of 30 nanometers. So from this distance to this distance is 30 nanometers. In the nucleosome, we saw it had a diameter of between 10 and 11 nanometers. So we go from this 10 to 11 nanometers to 30 nanometers. And here we see an electron micrograph that shows a 30 nanometer fiber. This would be seen in chromatin as well as the nucleosome level. This illustration from our textbook shows us how we go from the double helical level of the DNA all the way to the 30 nanometer fiber. So you see the DNA double helix here, and then you see the individual histone proteins. Notice that they have a tail, so you have a globular region and you have a tail region on each histone protein. 8 of those histone proteins come together to form the nucleosome core, and then the DNA double helix wraps around that core, making the nucleosomes. And the nucleosomes are about 10 nanometers in diameter, and there is some DNA that is not involved in that wrapping, that is called the linker DNA. And you see here how that looks. You've got that beads on a string look that we get from the nucleosomes. And then over here on the right, we see how the nucleosomes come together to make a denser and thicker fiber in which the DNA is going to be even further condensed than before. Now we go from the 30 nanometer fiber all the way up to the chromatid level. So before we saw the 30 nanometer fiber in which the nucleosomes interacted with each other. and then see how that 30 nanometer fiber is going to be organized sort of in a rosette kind of shape to form a bigger fiber called the 300 nanometer fiber. And that 300 nanometer fiber is going to form loops, and those loops are then finally going to form the chromatid, which has a width of 700 nanometers. This process is not completely understood yet. There's lots of active research going on about it, but basically we see that there are successive levels of increasing condensation as you go from one level to the next in the formation of the chromosome. And then the metaphase chromosome, like what we see here, is the highest level of condensation. This is when the genetic material and the proteins have reached the most extreme amount of folding and shortening that is possible with the DNA in our cells. Having a structure that is so complicated, sometimes we forget that each chromatid on a chromosome is only one long molecule of DNA. So yes, the DNA is associated with proteins, but if you could unwind it all, it would be one long uninterrupted molecule of DNA per chromatid. That molecule of DNA would be thousands of times longer than the diameter of the nucleus that it is packed into. So the condensing of the DNA first into chromatin and from chromatin into chromosomes allows the DNA not only to fit into the nucleus but to function in the nucleus as well. And like we said already, the highest level of condensation is represented by the chromosomes during metaphase. As far as genetic information is concerned, a chromosome is the same thing as chromatin. In other words, the chromatin contains the same DNA as a chromosome does, but just in a less condensed form. When we look at chromatin, you can observe within chromatin areas that are in the 10 nanometer nucleosome at that level of condensation. A lot of it is in the 30 nanometer level, and some of it is at the 300 nanometer loop level. In an interface cell, there is really no chromosomal scaffold. Remember, the scaffold is that protein structure on which the chromosome will condense, and that has to be put together every time the cell is going to go into mitosis or meiosis. Nevertheless, the chromatin isn't just thrown together in the nucleus. It is very organized, and it has been learned recently that the looped domains, those 300 nanometer loops, are attached to the nuclear lamina and the nuclear matrix. So each of them has a specific location in the nucleus. This picture shows us very nicely how the chromosomes and the chromatin are the same thing, and also that the chromatin is organized in the interface nucleus. So in the upper picture, we're looking at a spread of chromosomes, and each chromosome pair has been stained a different color. So one pair is light green, another pair is orange, another pair is yellow, and so forth. The lower picture shows a nucleus in interface of the same type of cell. And here we can see that those colors are not just all scrambled together, there are specific regions. So here is this light blue region, and here is this yellow region, and so forth. They do seem to be very specific regions that each, those chromosomes maintain their own spaces within the nucleus. If they were all mixed together, all those colors together might give you like some kind of like a brown color if it was all mixed together, but it isn't. You can see each of the colors separately in the cell. And then you see that this, you can envision how this could turn into that these chromosomes are really the same material as the chromatin that we see in an interphase nucleus. In general, chromatin is much less dense than chromosomes, but even within chromatin, we distinguish between more or less condensation. So there are two kinds or types of chromatin. Euchromatin is the least dense one, and it tends to contain the genes that are more active, genes that are getting transcribed. Then there is another kind of chromatin that is called heterochromatin. And heterochromatin tends to be more condensed, and it tends to contain genes that are not transcribed, so genes that are not active in that cell. And being more condensed, it is bound tighter, the DNA is bound tighter to the histone proteins, and therefore it's not accessible to the proteins that start the process of transcription. An example of heterochromatin includes our centromeres and telomeres.